All right, Chemistry 102, let's continue on with the final exam review. We're at question number 65. An alpha-1,4 glycosidic bond is found in what types of molecule in plants? And the answer is that type of bond is found in a starch, specifically amylose. It's also found in amylopectin. The amylopectin has branching too. Next one says, name 3 roles of lipids. I would accept a lot of different things here. For example, energy. Okay, we get a lot of energy from beta oxidation, which was the subject of chapter 23. Cell wall structure. I'd say another one is insulation. I mean, you could write down anything here. Hormone production. Okay. Energy storage. Okay. What are the components of a triglyceride? What type of functional group links these components? So a triglyceride is made from a glycerol backbone. If I was to, put hydroxyl groups here, this is called glycerol, right? But when we take a glycerol backbone, and then we attach a fatty acid to each one of these, okay, like this, then we get a triglyceride. So this is a triglyceride. And what type of functional group links them all together? Well, you can see that it is an ester, right? Here we go. Stearic acid is very common in both plant and animal tissues, while oleic acid is common in plant tissues. How does oleic acid differ from stearic acid? Well, I wouldn't expect you to memorize the structures of either of these carboxylic acids, but we can write them down and discuss them. So stearic acid has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. So 1, 2, there's stearic acid, and oleic acid has an unsaturation or a unit of unsaturation at carbon number 9. So up here 1, 2, 3, 4, 5, 6, 7, 8, 9. Then we have something that looks like this. 10, 11, 12, 13, 14, 15, 16, 17, 18. So this is oleic acid. And you can see the difference between these two is that stearic acid is a saturated fatty acid and oleic acid is unsaturated. And so that's the difference between the two of them, right? That's why oleic acid is more commonly found in plants, because we think of oils as being found in plants and saturated fats as being found mostly in animal tissues. Number 69, again, I wouldn't ask you to draw a phosphatidylcholine, but you should know that it's a phospholipid. It says what class of lipid is it? And it's found in cell membranes. So a phospholipid, found in cell membranes. Okay, and if we look at the structure of phosphatidylcholine, something looks like this. It's got a glycerol backbone, and then we have the choline head group, so we could just put that. So glycerol backbone, and then the choline head group. Draw the straight chain form of D-allose, the C3 epimer of D-glucose. Then draw the correct alpha ring form of this molecule. So look, let's start by drawing D-glucose. So we'll start there with, there we go. Okay, so we've gotten this far. This is D-glucose. An epimer is where we just reverse the stereochemistry. So 1, 2, 3. So we're going to reverse this stereocenter here. So when we draw the allose, it's going to look like this. So here we go. And then we're going to have 1, 2, 3, 4. So I have all the hydroxyls here, like that. So remember, everything that's on this side of the molecule is going to be pointing down. Right? These are all going to be pointing down in our Haworth projection. So let's start with this. And if you want to, you can number the carbons. So this is 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4, 5, and 6. At carbon 1, it says draw the alpha form. So that means our hydroxyl is going to be pointing down here. And at carbon 2, the hydroxyl is pointing down. At carbon 3, it's pointing down. At carbon 4, it's also pointing down. And sorry, at carbon 5, it's neither here nor there. So we can eliminate these ones that are down. At carbon 5, there we go. And so this would be alpha. This would be alpha D allose. That. What feature makes a carbohydrate reducing or non-reducing? We'll make it simple. We'll say non-reducing sugars. Non-reducing sugars do not have a free hydroxyl at the anomeric carbon. Glycogen is structurally very similar to which compound? That would be amylopectin. Similar to amylopectin. You know, they both have alpha 1,4 linked D-glucose molecules. They both have branching, 1,6 branching. The difference is the glycogen's branches are more frequent and shorter. So glycogen 1,6 alpha 1,6 branching is shorter and more frequent. Okay. Why are lipids insoluble in water? Well, that's, should be a question you know the answer to by now. They're mostly made of non-polar carbon-carbon and carbon-hydrogen bonds, right? And like dissolves like, and so since water is polar, a lipid is not going to dissolve in it. What polyalcohol is a component of all triglycerides? Give its common and IUPAC name. Well, we went over triglycerides this evening. You know that the answer is glycerol. Okay, so the name of the molecule is this, which is glycerol. The IUPAC name for it would be 1, 2, 3, right? You have 1, 2, 3 carbons. Propane triol. What's the minimum number of carbons in an alpha amino acid? It's 2. Is the minimum you can have, right? If you draw an alpha amino acid, right? This is the alpha carbon here, and if you have an amine, this would be glycine here. Okay, so the minimum number of carbons is 2 carbons. You got to have at least 2. From the list of amino acids below, identify the following structures. I would never ask you to memorize the amino acids, but you might have memorized some of them along the way, right? If you have just a methyl group as your R group, right, CH3, That's alanine. Phenylalanine is where you just replace one of these hydrogens with a phenyl. And so this is phenylalanine. Cysteine has a sulfur in it. So this is cysteine, right? Which, remember, it can undergo an oxidation reaction with another cysteine molecule to make cysteine, which has a disulfide bond in it. And serine has a hydroxyl in it, so this is serine here. Anyhow, you might have picked some of those up along the way, but if not, that's totally okay. What's this Vitter Ion? It's a molecule with both a positive and negative charge. For example, if you look at glycine at physiological pH, it's going to look like this. Right, we got a positive charge and a negative charge. Draw the structure of this amino acid below a pH of 10. This is lysine. pH of 1, sorry. This is lysine. Okay, so a pH of 1 is extremely acidic. And so what's going to happen, right? Amines are bases, and so those will accept a proton in a carboxylic acid. Well, that's just going to remain intact at a pH of 1. And so lysine will look like this. We have 1, 2, 3. I'll draw a wedge here. So we'll have our protonated amine here. We'll have our protonated amine here. And the carboxyl group will simply remain intact. How many amino acids have acidic side chains? It's another one. I wouldn't expect you to have this memorized, but maybe you picked it up along the way, because we did look at, polar groups, acidic ones, basic ones. The answer is there are 2. Okay, there's aspartic acid, and glutamic acid. Aspartic acid it has 1 CH2, and glutamic acid has an extra 1 in it. What else? Draw the hydrolysis reaction of Alanyl phenylalanine. Well, first you'd want to draw the amino acid. So the first one is an alanine, and we'll just start by drawing NC carbonyl, right? And then we have NC carbonyl. So the first one is alanine, so it's going to have a CH3 group. The next one is phenylalanine, so it's got CH2 and then a phenyl. Let's fill out the rest. We've got hydrogens here, and this nitrogen will have a hydrogen. The bond that we're going to break is the peptide bond, or the amide bond. So this is peptide bond, and this dipeptides, we're going to cleave that bond. And so we'll hydrolyze that in water. And then what are we going to be left over with? We're going to be left over with alanine. So let's draw that. Left over with alanine as this Vitter ion, like that. And then we'll also be left over with phenylalanine. Just condense that a little bit. There we go, CH2. I'll draw the whole phenyl because I got room. There we go. How many atoms form the backbone of the repeating unit of the polypeptide? Well, the answer is 3, right? Because you always have NCC, right? I taught you that enough so that you should know that it's 3 atoms and then another 3 atoms and then another 3 atoms in a peptide. What are the 2 most common forms of protein secondary structure? That would be the alpha helix and the beta pleated sheet, both which maintain their form by hydrogen bonding. When naming a polypeptide chain, you begin at the blank and go towards the blank. We never talked about naming polypeptide chains, really. It doesn't come up in the ACS final, so I don't ask any questions about it on your file. But you should always start at the left-hand side. So you start at the N terminal, and you go towards the C terminal, or as I call it, the N terminus to the C terminus. The identity of the R groups contributes most significantly to the blank structure of proteins. That would be the tertiary and quaternary structure. tertiary. Let's just leave it at tertiary. It could also be the quaternary. Quaternary. All right, because remember that when you have the tertiary structure of a protein, all the nonpolar R groups are going to be pointing in towards the center of the protein, and then all the polar R groups are going to be pointing outside towards the aqueous media. Explain why soap would effectively denature a protein. I'm just going to kind of talk our way through this one. I could write it down, but that'll eat up a lot of time, and I have the answer posted to our class shell. But what I wrote down here is soap has a hydrophobic and a hydrophilic side, right? If you think about what soap is, it's just a carboxylate of a fatty acid, right? You take a fatty acid, okay, and that's essentially a soap. Okay, so we have a water-loving portion, and then we have a water-hating portion here. And so proteins have both hydrophilic and hydrophobic portions too, don't they? So the soap can be attracted to those groups, and then it's going to force the protein apart, right? Because again, as I told you, the tertiary structure of a protein, we're going to have the nonpolar groups on the inside, and this portion could interact with that, and then the polar groups will be on the outside, and the hydrophilic portion could interact with that, and it would ruin or break those intermolecular forces that hold the tertiary structure of the protein apart. Okay, it's asking us to draw some amino acids here. So 2 aminopropanoic acids. So if we start by drawing propanoic acid like this, and then we count 1, 2, and we put an amino group here. Okay, so that one's done. And it's asking us to draw 2 amino 3 4-hydroxyphenyl propanoic acid. So let's start by drawing 1, 2, 3 propanoic acid like this. So on the 2, we're going to put our amino, and on carbon 3, which is this here, we'll put 4-hydroxyphenyl. So 4-hydroxyphenyl is just this. Okay, you have a hydroxy in the para position, and so we'll put that. Yeah, sorry, I'm adding an extra. There we go. Okay. Then it's just asking you if they look familiar to you. And the answer is yes, they should look familiar to you. Again, I don't ask you to memorize these, but this is alanine here. And then this one here is tyrosine over there. What are 3 roles of proteins in the body? I don't know. I wrote down a few different things here. I put enzymes, of course. Enzymes. I put transport, involved in oxygen transport, like for example, hemoglobin and myoglobin. And I also put antibodies, hormones. Okay, there we go. Come on. Number 86, explain what's wrong with the following statement. Enzymes shift the chemical equilibrium to more heavily favor the products. Well, that's not true, right? If you have a reaction like this is your reaction in time. And if you have the energy here, if you have your starting material here, and you have a certain activation energy associated in order to make your products, right? This energy barrier here, that's the energy of activation. But if we run an enzyme catalyzed reaction, that's going to change the pathway, but it also lowers the activation energy. So the equilibrium remains the same, right? It has nothing to do with equilibrium, okay? Enzymes do not affect equilibrium. They increase reaction rate. Reaction rate only. Okay, by lowering the energy of activation. Okay, number 87, still on enzymes. It says in competitive inhibition, does the inhibitor bind to the active site or to an allosteric site is this an example of reversible or irreversible inhibition? Well, if it's competitive inhibition, the inhibitor is going to bind at the active site, and that's going to be reversible. And the classic example here would be a drug. Right? A drug is going to bind in an active site, but we know that the drug, if you take a drug, it's not going to be in your system for the rest of your life. You're going to metabolize it and it's going to leave. Why? Because it's a reversible binder. Number 88, what molecule is primarily responsible for conserving the energy released in catabolism? That would be adenosine triphosphate, or ATP. What is the most readily used energy source in the diet? Not what gives you the most energy, but what's the most readily used? And the answer would be carbohydrates. Carbohydrates, those are most quickly used up. Glucose, right? You can get more energy from the fat, but carbohydrates are more readily used. What are the end products of glycolysis? Well, we spent quite a bit of time looking at glycolysis. The end products are you end up with a pyruvate, ATP, and NADH, and you have two pyruvates, two NADHs, and two ATPs. When an enzyme has the term kinase in the name, what type of reaction do you expect it to catalyze? And the answer would be a transfer of a phosphate group, right? We looked at kinases and we said the name kinase isn't intuitive, but that's what kinases do. Define gluconeogenesis and describe its role in metabolism. So gluco means glucose, neo is new, and genesis is making. So it's a pathway that results in the synthesis of glucose from non-carbohydrate starting materials. So synthesis of glucose from non-carbohydrate starting materials. Okay. What does the term hypoglycemia mean? This is low blood sugar. Low blood sugar. I remember this question was on the last ACS final that I gave. So deficiency of glucose in bloodstream. And of course the opposite would be hyperglycemia. What effect does insulin have on blood glucose levels? Well, it lowers blood sugar levels, doesn't it? I told you I had a friend, when I first moved to the States, we'd go to the In-N-Out or whatever. What was it? Five Guys Burger and Fries up in Boulder. And before we'd walk into the restaurant, he'd take his insulin pump and he would crank it up. So insulin lowers blood sugar levels by absorbing glucose, and converting it to glycogen. What molecule serves as a signal to increase the rate of reactions of the citric acid cycle? Holy hell, you could put anything here. I mean, you could put ADP, you could put adenosine monophosphate, right? Because you are have low ATP, and so any of the products of the removal of a phosphate from ATP. Next one, what molecules serve as signals to decrease the rates of reactions of the citric acid cycle? Well, if you have a high level of ATP, you have a high level of NADH, and if you have a high level of FADH2, those will all decrease the rates of reactions of the citric acid cycle. Number 97, what is the terminal electron acceptor in aerobic respiration? The answer to that is oxygen, right? You take two protons, right? They get pumped across the matrix space. They add with oxygen, and then you combine those two electrons, and what do you end up forming? You end up forming water. So H2O. How many cycles of beta oxidation can myristic acid undergo? How many FADH2s will be produced? How many NADHs will be produced? So myristic acid has a total of 1, 2, so it's got 14 carbons. So let's write out the structure of myristic acid. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. It can undergo beta oxidation. Look, 1, 2, 3, 4, 5, 6. Do something up here. 6 times, yeah. So can undergo 6 beta oxidation cycles, and then it's going to produce 7 2-carbon units, right? Look, you've got 1 here, 2, 3, 4, 5, 6, 7 2-carbon units. So that means you're going to make 7 FADH2s, and you're going to make 7 NADHs, okay? That's all it's asking us for. What enzyme hydrolyzes sucrose? The answer is sucrase, right? Enzymes usually end in A's. What enzyme hydrolyzes maltose? The answer is maltase. Okay, name the 3 types of molecules, or name 3 types of molecules that would be classified as lipids. My favorite is waxes, steroids, triglycerides, GCs. What else? Glucotrienes, glucotrienes, PGs, prostaglandins. Okay, those all would be. What else? Sphingolipids. Sphingolipids, right? Why is an ester like methyl acetate water soluble, whereas an ester like myrisyl cerotate down in beeswax not water soluble? Well, if you compare their structures, this would be methyl acetate, In myrisyl cerotate got much longer chains. Now I wrote down the condensed structure, but you've got 26 carbons on the acid side, and you've got 30 carbons on the alcohol side, right? You've got 30 carbons over here, and you got 26 carbons over here, so 30 carbons. Okay, so that's a total of what? 56 carbons. Well, it's just based off of the fact that the hydrophobic chains in myrisyl cerotate are so much bigger. So we'll just put here, let's make it simple because we're running out of time. Larger hydrophobic chains. Nothing more than that. And the last one is what kind of linkage connects these molecules together? We can see that this is pointing down. So this is alpha. And then if you count the carbons, look, you have 1, 2, 3, 4, 5, right? So it's 1 and 4. So this is alpha 1, 4 linkage.